Hello, everyone. My name is Patrick Akwaka. I am a professor of medical microbiology here at the Faculty of Medical Sciences. We are of the unit of pathology, microbiology in the Paraclinical Sciences Department of the University of the West Indies, St. Augustine campus. It is my distinguished pleasure to welcome you all to the series of recording and taping of our laboratory diagnosis of infection. We want to expose you to what we do in the lab. And in the unit, we do have the academic staff, Dr. Kuhade, Arvin Kuhade, and Dr. Chandra Shikha Unika. And then we do have the laboratory technicians, Mr. Steve Larman and Mr. Renan Thompson. We would want to present these videos to you, which is exactly what we'd expect you to come into the lab to do. There's going to be a series of recordings. You're going to be taken through how specimens are collected for you to know the biosafety measures you should concern yourself when you come to the lab. We'll also be taking you through how we work out several specimens in terms of trying to determine what is the diagnosis in urinary tract infections, in urogenital tract infections, in central nervous system, blood system, respiratory system, gastrointestinal system. So we really would want you to pay close attention to the details of what will be explained to you. And in that way, you would really be assured you're not going to miss anything. So once again, we want to welcome you, and we want to also use this opportunity to thank the members of CMSC staff. So we are very glad to have you, and we wish you the best and good luck. Thank you.
Good day, students. My name is Steve Lalman. I'm one of the microbiology technicians at the Department of Paraclinical Sciences, Faculty of Medical Sciences, University of the West Indies. And today, we are here to talk about the laboratory diagnosis of infections in the microbiology laboratory. Now, I remember a great physician said, 3 things you need to do for proper diagnosis. 1 is a proper history, 2, a physical examination, and 3, the appropriate investigations. And such appropriate investigations will probably most likely involve a sample in the microbiological or microbiology lab. So today, we're going to talk about different aspects in terms of how do we actually diagnose an infection in the microbiology lab. And the subtopics would include, first of all, you have specimen collection and transport, you have microscopic analysis, and also a macroscopic analysis. You have cultures, you have identification using biochemical tests and other means, sensitivity testing, and your final report. Now, in the microbiology lab, infections are diagnosed or infections are identified using several ways. The gold standard is culture, but sometimes culture may not work, depends on the etiology. You may have to resort to molecular biology or DNA testing for identification of such pathogens. Now, it is very important for microbiological samples that specimens are collected at the appropriate time, ideally before the administration of antibiotics. The amount of specimen is very important too, and the type of specimen will all influence the quality of the results. So now, we're going to get into the actual specimens, the different types of specimens you may encounter in the microbiology lab. Probably the most common sample you will encounter is a urine sample. All right? In later videos, we will get into the actual processing of a urine sample. This is just an example to show you what a urine sample looks like. You will also have sterile specimen containers that is used to collect a number of other samples. For example, maybe a urine, as we said, maybe a stool sample, maybe a sputum, etc. Please note that the sample must be labeled properly after collection as to the nature of the specimen, the date, position, etc., etc. Other samples you may encounter is the collection of blood for blood cultures. Blood cultures are very important, and they can be collected in different vials or pairs or sets of vials, which we will discuss in a later video. Also, you can have what you call culture swabs. These swabs are culture swabs that are used to collect specimens, for example, throat swab, sample from the skin, nose, any wound swab, and so on. And they actually are made with a large cotton tip-like applicator and a transport medium where you place the sample, the swab, after collecting the sample and send it off to the lab. Ideally, samples will be transported within 1 to 2 hours to the microbiology lab for proper yield of the etiologic agent. So one of the first tests we're going to talk about is the catalase test. The catalase test, the principle involves the breakdown of hydrogen peroxide into oxygen and water by the enzyme catalase. Some bacteria possess the enzyme catalase, for example, your Staphylococcus bacteria, and some do not, for example, your Streptococcus bacteria. So we're going to demonstrate now how to do your catalase test. So what you'll need is a tube, a test tube, where we place some hydrogen peroxide for safety. This test can also be done on a microscope slide. And we're going to pick up some of our bacteria, our Staphylococcus bacteria here, using a wooden applicator stick. And then we're going to immerse it in a test tube with your hydrogen peroxide. So here we have immersed it, and if you look carefully, you can see bubbles being given off from the reaction. So again, the reaction is the enzyme catalase, which is possessed by the bacteria, breaks down hydrogen peroxide into oxygen and water, and the bubbles you're seeing is the liberation of oxygen. So that was your catalase test. So looking at the reaction for the catalase test, I have used an agar plate, just a colored plate, just as a background to show you the liberation of the bubbles for easier viewing during this video. So the previous reaction you saw was a positive catalase test using the bacteria Staphylococcus. Now we're going to use a negative bacteria or a negative reaction, showing you where we're going to use a member of the Streptococcus genus. So again, we're going to take our hydrogen peroxide into our tube. Then we're going to pick up some bacteria of the Streptococcus species. Now one has to be careful when picking up the bacteria from a blood agar plate because some red cells will have the enzyme catalase. So when you pick up the bacteria, make sure you don't scrape the agar as well. So now I'm going to immerse my bacteria into my tube, hydrogen peroxide, and we look for a reaction. In this case, you see no reaction, no bubbles being produced because the bacteria here is Streptococcus, which does not possess the enzyme catalase, and thus you have no bubbles being produced. Again, the background I've used here is just to show you the tube and the hydrogen peroxide. We're going to demonstrate the test called the coagulase test now. Now, having done the catalase test, the catalase will differentiate between bacteria that are catalase positive and catalase negative. For example, your staph will be catalase positive and your streps will be catalase negative. Once you have a genus as a Staphylococcus bacteria, you want to speciate that bacteria in terms of what species of Staphylococcus you have. For example, you could have Staphylococcus aureus, you could have Staphylococcus epidermidis, or Staphylococcus saprophyticus. And the coagulase test will be able to differentiate between those 3 species. So now we're going to demonstrate how we actually do the coagulase test. So to demonstrate the coagulase test, what we'll need is some coagulase plasma, and we'll need our bacteria. So we're going to pick up an inoculum of our test bacteria here. and inoculate it into our tube with plasma. Discard in a biosafety container or biohazard container. And we have our bacteria here with our plasma, coagulase plasma, and then we're going to place this in an incubator for up to 4 hours incubation at 37 degrees Celsius, and then look for a reaction within that time. So we're going to place this in our incubator at this point. After incubation, we remove our tubes from the incubator and we look for a reaction. The enzyme coagulase converts fibrinogen to fibrin, and the species Staphylococcus aureus possess that enzyme, and thus we expect to see a clot, because fibrin will reveal a clot. So here we have 2 tubes that has been incubated, one positive and one negative. The positive one, Staphylococcus aureus, we tilt the tube and we see a clot has been formed. Notice if I tilt the tube, there's no liquid here, it has formed a solid clot. That's for Staphylococcus aureus. Staphylococcus epidermidis now, here we have, you can see it's a negative reaction where the plasma did not clot. Again, because of the bacteria not possessing the enzyme coagulase, so we have the plasma remaining in its liquid form. So that's the coagulase test. We will now demonstrate the oxidase test. The purpose of the oxidase test is to differentiate between 2 groups of bacteria of the gram-negative rods in terms of mainly the Enterobacteriaceae. Some that are oxidase positive and some are oxidase negative. So one common bacteria that you would find the oxidase enzyme being present is Pseudomonas aeruginosa. One that is negative for oxidase will be your common example of Escherichia coli, or E. coli as we commonly know it. So the principle of the oxidase test is to look for the enzyme, the cytochrome C oxidase enzyme. To demonstrate that, the reaction consists of using oxidase reagent, which sometimes comes in these little ampules. And this oxidase reagent is named tetramethyl p-phenylene diamine dihydrochloride. So the reaction or the method of the oxidase test is to take a piece of filter paper and add 1 drop or 2 of your oxidase reagent to wet the paper, and then we'll use either a loop or a glass slide or some object to pick up some bacteria and smear it on. The reaction you're going to see will be a color change where this oxidase reagent is broken down to a color which is reduced to a blue or purplish blue end product. And it happens within 10 seconds. So we're going to pick up some bacteria here. This is Pseudomonas aeruginosa. And then we're going to smear it onto our filter paper. And if you notice, the reaction changed very rapidly after it took a few seconds. It went from colorless to a purplish blue color. I'll repeat that again so to show you. So pick up some bacteria of Pseudomonas, smear it on, and we see the reaction developing there right before your eyes, that bluish color. So this is a positive reaction for your oxidase test. Now we're going to do a negative reaction, a different bacteria here. Here we have E. coli. I can smear it on to another spot of the filter paper, and you see no reaction taking place there, no color end product. Again, I'll repeat that for your E. coli. Pick up some bacteria, smear it on to a different portion of the filter paper that is wet with the reagent, and you have no reaction taking place. So to summarize for this oxidase test, a positive reaction is done by Pseudomonas aeruginosa and negative by E. coli. So now, folks, we're going to talk about the microscopic analysis. And using this, with this topic, you can do 2 things. Once the sample comes into the lab, you need to examine it to see what you have under the microscope. And 2 of these things or tests we can do is, 1 is a gram stain, and 2 is a wet preparation. So certain samples will require both a gram stain and wet prep, like vaginal swabs, whilst other samples may just require a gram stain. The gram stain has a 3-fold function. It classifies bacteria into 2 major groups, gram positive and gram negative. It also displays cell morphology in terms of the bacteria. Are they cockeye, round in shape? Are they elongated like rods? Are they spherical, spiral, etc.? And it also displays the arrangement, whether they are arranged in pairs, clusters, or chains. Also, under the microscope for a gram stain, you can identify white blood cells, red blood cells, and so on. White blood cells are important, which they normally indicate infection rather than colonization of that patient. So we're going to talk about two things now. We're going to talk about the wet preparation first, and then we'll move on to the gram stain, how to do a gram stain and make a smear. The wet preparation is done for several samples. For example, when doing a urine sample, the physician may request a urinalysis test and a culture. Under the urinalysis test, you will have two parts to it, a dipstick or chemical analysis, and a portion of the urine sample is spun down in a centrifuge tube, which I have here, and you can see what that looks like. So the wet preparation, like I said, could take many forms. One form of wet preparation involves doing that test for a urinalysis where we look at the sediment under the microscope. So would have poured some urine sample in this tube, centrifuge it for 5 minutes at 1500 Gs, and then look at the sediment under the microscope. So for urines, we will have sediment here where we're going to do a wet preparation of it. So it simply means mixing up the urine sample, using a pasta pipette to remove a drop of two, and place it on a microscope slide where we're going to apply a cover slip or cover glass to it. So here we take a cover slip or cover glass, apply it onto the slide, and then we go to the microscope and examine it to look for cells, namely white cells, red cells, casts, bacteria. Sometimes you can see bacteria, you can see candida, you can even see trichomonas, etc. under this slide. So this is one form of a wet preparation. Another form of wet preparation involves where you have vaginal swabs and you want to make a wet preparation to look for candida and a parasite called Trichomonas vaginalis. So you'll take a microscope slide, add a drop or two of saline onto the slide, take your sample, which would have come from the patient, and then emulsify it. Emulsify your swab on the saline. And then apply a cover slip or cover glass again to it to create that, what we call a wet preparation. And again, we put this under the microscope under times 10, times 40, and look for things like I said, like, for example, Trichomonas, candida, and so on. A third method or third type of wet preparation involves something called an indaying preparation, which we'll demonstrate when we're doing CSFs. So the indaying preparation is also another form of a wet preparation. Now we go on to actually doing a gram stain. The first step involves is making a smear of your specimen. So what you'll need is a microscope slide. There's a frosted side you want to label, and you can use a lead pencil to label it, because sometimes some markers will erase during the staining process. So you label your slide with a lead pencil, and we also use a wax pencil or grease pencil to make a ring circle on the slide. Sometimes, if the slide is not easily marked, you can warm it a bit with a flame, Bunsen, and then put it flat down, again make the mark with a wax pencil, where you get a nice circle on the slide. That circle helps keeps the stains item within a space and doesn't use up too much stain. So we have our slide ready, we have our bacteria, and we have our tools of the trade called our microbiological loops. These loops are metal loops, so you need to flame them and cool them before use. So we apply it to the Bunsen burner. Normally apply it to the cool part of the flame first and then bring it up to the hot part where it becomes hot red. Once it becomes red, you pass it across the entire length, the shaft of the loop to make sure it's sterile. Again, when doing these demonstrations or these tests, it's not advisable to be speaking because droplets could land on your slides and so on. But of course, for demonstration purposes, I am speaking while I'm doing the test. So we have our loops that are flame and cooled. We're going to add a drop of either water or saline onto our microscope slide. After 5 seconds, 10 seconds, the loops are cooled. So here we're going to take a drop of our saline, place it on our slide. And you want to handle tubes aseptically. So the proper way to handle tubes and cultures and so on, let's say, for example, you're going to open this tube. You're going to open it like this with the small finger of the other hand, flame the top, go into it, remove your liquid, place it on, flame again, and cap it back like this. That's the proper technique to handle your tubes when working in microbiology lab. After using the loop, of course, you must flame it again before putting it back down, because you don't want to use a loop that has bacteria or any other substance on it. And now we use 3 loops or so when one is cooling, the other ones would have cooled already. Now we're going to add our bacteria to our slide. So I'll use a plate here as a background to show you the color, to show you some contrast. Use a cool loop, and we're going to pick up a colony of bacteria. Now bacteria would have been streaked previously on display to give you colonies. If anytime you're unsure that the loop is not cool, you can just touch the loop on a clear part of the agar plate to make sure it's not hot, and simply touch one colony, which is one of these little dots, to pick up to do your gram stain or make your smear. Then we're going to apply this to your slide. I'll turn it sideways so you can see it, and just emulsify it a few times within that ring circle. That's it, a few swirls you need. Spread it out on the slide, and then of course flame back your loop and place it down to cool. So when you make your smear, I'm not sure if you can see this properly, but you'll see the suspension being very light, a very light milky suspension. You don't want it too heavy or too dense. If it's too thick, it will affect the staining process and the outcome. So there's a light suspension you need to create, a suspension, and once that is made, you can simply place it next to your flame on the bench, and the warm draft will dry that slide. It's important to air dry the slide before moving on to the staining process. Once the slide is air dried, you need to go on heat fix by passing it through the flame, which I'll demonstrate in a short while. So while the slide is drying, in preparation to doing the gram stain, I will now move on to the culture to show you how we culture agar plates, how we streak, etc. In our demonstrations, we're going to demonstrate now how to perform a culture in the microbiology lab. Now you can have cultures of various specimens. For example, you can have a throat swab, a skin swab, wound swab, etc. Come into the lab to perform a culture. Or you can have bacteria that was previously cultured and streaked on agar plates that you want to do a subculture onto fresh agar plates. Now agar plates that has not been inoculated come in a variety of forms for different specimens and so on. Here we have a blood agar plate, nicely bright red in color, that is made from sheep blood, animal blood. It's the best type of blood to use for our blood agar plates. Here we have McConkey agar, which is used to grow your gram-negative rods. We have clear agar, a nice greenish color that's used for the culture of urine samples. And also we have a nice brown agar called chocolate agar used for fastidious bacteria like Neisseria, Haemophilus, and so on. These chocolate plates need to go under special atmospheric conditions. Carbon dioxide, about 5 to 10% carbon dioxide, which is achieved in either a candle jar or a CO2 incubator. The purpose of using different plates is because you have different pathogens needing different growth requirements. Your blood agar plate will grow generally all types of bacteria, whether it's gram-negative, gram-positive, and so on, whilst your McConkey and Clear are mainly for the gram-negatives. Clear can grow both gram-positive and gram-negative, so it is a multi-purpose plate. So I'll demonstrate now how to do a culture using a sample of a swab. So let's say the sample came in, again, maybe a wound swab, skin swab, throat swab, and so on. In that case, all we will need is a blood agar plate for throat swab. So I'll demonstrate, let's say, for example, a throat swab. So you'll take an agar plate, and you will label your plate, in top we normally write the specimen. So if a throat swab, we will write TS. We'll write the sample number and our date. Right. Once the plate is labeled, and you must label your plate before you actually place the specimen, then we'll take our sample, in this case our throat swab, and we're going to make a pool or inoculum into a small portion of the plate. So we will turn the swab, rotate the swab while making that circular inoculum on the plate. A small, very small portion. Place back the swab into its container. That inoculum now we're going to use to what we call streaking. Now streaking is a technique used to dilute out your concentration of bacteria. So we have our inoculating loops again, which have been flamed and cooled before. And now we're going to show you how to streak the agar plate. So streaking, again, will create colonies the next day. So we're going to take our loop again, and we're going to start off with the pool and pull out about 4 or 5 times from the pool onto a fresh agar or fresh part of agar. This part is especially important in terms of contamination. Namely, again, when streaking a plate, one must not be speaking, so again, droplets can land on the plate. But of course, for demonstration, I'll be narrating throughout the process. So we're going to streak our plate in what we call quadrants. So I've done the first quadrant. So I'm going to use another loop now, cooled, and go back to the previous line of streaking and pull out about 3 or 4 times, and then a couple times outside that section. Keep the plate covered while flaming and cooling, so to avoid any contamination. I'm going to go now to the third quadrant, pull out again from the previous streak, and I can turn my loop over and do the fourth quadrant like this. All right. So that is using your loops to streak a plate. Now, there are different varieties of streaking method, many different techniques one can use, but the main thing you want to do is try to streak to achieve the end product like this. So this is our streak plate. We're going to place it in the incubator. So all plates are incubated at 35 to 37 degrees, 18 to 24 hour growth. The next day we remove those plates, and hopefully we can see an end product, something like this here, where the streaking would have revealed dilution of the organisms to create what we call colonies. So what would have happened overnight is where one bacterium was moved to the plate, that one bacteria multiplied exponentially to create hundreds of bacteria or thousands on that one spot. So every single spot here is what we call a colony. And those are colonies we need to use in the lab to do our different tests, etc. Sometimes if you don't have access to a Bunsen burner in a lab, you will have disposable loops where you don't have to flame and cool. These disposable loops are sterile, and I'll show you how to streak a plate using a disposable loop. So again, we'll take an agar plate, we'll label it with our sample name, the type of sample, sorry, so like for example, a throat swab, our specimen number, and our date. Of course, we have a bacteria we're going to use, or we can, let's say if it's a throat swab, we will use our specimen source here again. So we have our throat swab here. Again, we're going to open the plate and make an inoculum, rotating the swab, making a small pool. You don't want to make the pool too large because you're going to use up too much space for your agar plate. Once the inoculum is made, insert back the swab, and then we go on to streak with our disposable loops. Loops come in different sizes, they all color coded, etc. Again, this is sterile, you don't have to flame and cool. Simply open and you streak your plate by pulling out 3 or 4 times from the pool. And then you can turn the loop over the other side and streak out that first quadrant. put it in your biohazard container for disposal. You're going to use another loop. And again, you pull out that second quadrant, turn the loop over, pull out the third quadrant, and spread it out over your agar plate. Dispose of your loop. So notice how fast that was. No flaming and cooling. It's very fast, and by streaking using a disposable loop. So now we're going to speak about inoculation using for biochemical tests or biochemical tubes. As part of the investigation, having done your culture and come up with a growth on your plate of a suspected pathogen, you now have to try to identify this bacteria. And this is done by doing several things. For example, the gram stain, maybe preliminary tests like catalase, coagulase, and so on. And for your gram-negative rods, like members of the Enterobacteriaceae group, you'll want to use these two biochemicals. And we have some examples here which are going to show you how to inoculate. So these two biochemicals will be, one of the tests we can do is called a TSIA, which stands for triple sugar ion agar. We also have a citrate test, motility test, etc. So I'm going to show you how we actually inoculate these tubes to get our growth reactions to help us identify the bacteria. What we'll need is another tool called a straight wire. This straight wire is done for inoculation. So again, you want to heat the wire before. And in this case, you must heat the entire length of the wire shaft to make sure it's sterile. Within 5 seconds or so, it will cool. While that is cooling, you want to make sure and label your tube, so you can make sure and label them before with the specimen type, specimen on a plate, which bacteria you're going to identify, and the date. All right. So we'll label up our tubes quickly. The best labeling tool you can use is a Sharpie marker. Sometimes the wax pencils will erase easily. Sharpies works fine. Label them. Now, because some time has elapsed, and to avoid contamination, you want to make sure and have your straight wire again sterile. So I'll go back and flame again. Again, you're not supposed to be speaking while doing this test, but again, for demonstration purposes, I'll be explaining through the procedure. So the loop will have been cooled within 5 seconds, and we're going to touch one colony. Now, a good trick or tool to remember is that you must work with one colony throughout the process. So you choose a representative colony, and you just take a little touch there. You may not see it, but once you see the colony be moved, means you would have picked up bacteria. Again, using standardized technique for handling your tubes, what they call aseptic technique. Open the cap with your little finger, pinky finger, flame the top, and then going straight, keeping your hand steady. At the beginning, your hand may shake, but that's okay. And then we're going to inoculate the tube all the way down the center of it. And then when we withdraw in, we're going to streak it across the slant of the tube. So this technique for the TSI is called a stab and a streak. So you would have stabbed it in the beginning and then streak the slant. Cap again, flame and cap. Without going back to the bacteria, there's still bacteria here on the straight wire. We go on to our next our tube called the citrate. Again, straight down. This one we don't stab, we just streak. So we're just going to streak this slant. And so we go on inoculating your different biochemical tubes, whether it's a stab or streak or both of them. After we have inoculated, you want to take all your biochemical tubes. The very important one you must leave slack for air to pass through is your TSIA. So make sure it's not covered tightly. There must be a space for air exchange. The others can be tight. And you place these in your incubator overnight for growth again. The next day, you're going to remove them, and hopefully you'll see some reactions like these here. These are positive reactions of bacteria that a TSIA have reacted. So this is a TSIA that is, let's say, that is negative or without bacteria, that shows no reaction. These are ones that have bacteria, showing you the end products. Right? Then we'll go to a chart. We have what we call identification chart, and list these reactions and compare the reactions to distinguish what bacteria we have, namely members of the entire Bacteriaceae group, like E. coli, Salmonella, Shigella, Klebsiella, and so on. Those are the bacteria we use these biochemical tests for. So that's the end of biochemical inoculation. So to continue the video for how to make a gram stain, we have done preparing our smear. Remember, we place our bacteria on our slide and we let it to air dry. It's very important to let it to air dry to make sure it's completely dried. How do we check to see if it's dried? You can look at the smear, put it against a background to make sure you see a nice film on it. There is no droplets, nothing wet on it. Once it's dried, then you're going to move across to heat fixing. Now heat fixing is done. We're going to pass the slide from the bottom, the flame at the bottom, 3 times through the flame to allow the bacteria to adhere or stick onto the slide. So when you stain it, it doesn't wash off or come off. So you're going to go 1, 2, 3. 3 times through the flame. Touch it at the back of your fingers or hand. It should be just warm. You don't want to do too much heat, just gentle heat. And once that is done, the slide is now heat fixed, and we're ready to go across to the staining station now to stain for the gram stain. Right, so we're going to continue with the gram stain procedure where we have made a previously heat fixed smear. We're going to take our slide or smear and place it on a staining rack, and then we're going to apply our staining reagents, which there are 4 of them. So you'll need our staining reagents. We will need some distilled water in a squeeze bottle, and we're going to need some blotting paper called bibulous paper for drying the slide. The staining procedure will consist of using these 4 reagents. The first one is crystal violet. The second one, iodine. Third one, your decolorizer, which consists of acetone alcohol mixture. And the fourth one, safranin or carbonyl fusion. The timing now involves normally 1 minute of crystal violet, 1 minute of iodine, a few seconds of decolorizer, and 30 seconds of a counter stain. So now we're going to apply the staining stains to your slide and try to explain the principle of what happens while the gram stain is being done. So I'm going to apply a drop or 2 of crystal violet. Make sure the bottle is open. You don't have to squeeze, just invert, and a drop or 2 will come out from the spout, and it will fill that ring circle. And that's the purpose of making that ring in the beginning when we're making our smear. So it contains the stain. So now this has to sit and react for 1 minute. So in the slide, we have placed 2 types of bacteria. So I want you all now, using the principle of the gram stain, for you to think about what happens at the cellular level as the stain is reacted. So we have 2 groups of bacteria, gram positive and gram negative. At this stage, the color of the cells, these both types of cells, gram positive and gram negative, will take up the purple bluish color of the crystal violet. After a minute, we will have moved on to your iodine. The purpose of the iodine is it acts as what we call a mordant or stain enhancer that enhances that color of your crystal violet. It actually forms a complex with your crystal violet within the cell wall of your bacteria. What you should remember from your theory is that the cell wall structure for both gram positive and gram negative is very different, in that the gram positive wall has a very thick layer of peptidoglycan, which is a key layer for keeping that complex color inside. So a minute would have been passed or about to come up. We're going to decant now our stain into the sink or staining tray and simply rinse off with distilled water. Now when using distilled water, don't squirt on the smear itself. You squirt on the top of the smear slide, and the water will run down to wash off that excess stain. All right, so you squirt around it, not on the smear itself. Shake off the excess. It doesn't have to be completely dried. And now we're going to add a drop or 2 of your iodine. Again, this must sit and react for 1 minute. So again, think about the reactions happening here now. So the iodine, as we said, it forms a complex with that crystal violet within the cell walls. And at this stage again, both gram positive and gram negative cells are appearing purplish color if you were to view it under the microscope. So assuming a minute has passed for time, we're going to decant again and wash off that iodine. Again, do not squirt on the smear itself. Shake off the excess. Now we move to the third stage, which is the most important stage or step of your gram stain, your decolorizer. Decolorizer or decolorizing takes sometimes a few seconds. depending on the thickness of the smear. And that's why it's important to have a very thin smear when you're making your smear at the beginning of the procedure. Now, to decolorize, you cannot put the slide flat or leave it flat. You have to hold it upright in this angle and apply the colorizer drop by drop until the blue color washes off. And it just takes sometimes a few seconds. So hopefully you can see this. I'm going to apply drop by drop of your decolorizer. Once the blue color stops coming off, it runs clear, you stop that, and you apply quickly some distilled water around the smear again to stop that decolorizing process. The reason why you have to move very quickly is because you can over-decolorize a smear by adding too much decolorizer and forcing that color out. So now we are at the third stage. We just completed the third stage, and I want you to figure out what's happening at the cellular level now. Remember, we had two types of bacteria here, gram-positive and gram-neg. The gram-positive cells would have retained their bluish-purple color of the crystal violet and iodine complex because it has a thick layer of peptidoglycan. So that complex is very tight, it cannot be easily washed off. So the gram-positive cell remains bluish-purple. The gram-negative cell now, however, would be colorless because our decolorizer would have washed out that complex within the cell walls. And that is because it has a thin layer of peptidoglycan as its cellular wall structure. So the gram-pos now would have been purplish. The gram-neg would be clear in color. We go on to the final stage now, which is our counter stain or saffronin. So again, we're going to apply a few drops. Let it react for 30 seconds, and then we'll wash off and dry. So assuming 30 seconds has been passed or elapsed, shake out again the extra stain, wash around the smear. Sometimes you can wash the back of the slide too to get a nice clean slide, and shake off the excess. So now we have the gram stain that is finished here. And at this stage, I want you again to imagine or figure out what is the color of the cells before you go to the microscope. Again, the gram-positive cells would have retained that bluish-purple color because of the thick layer of peptidoglycan, whilst the gram-negative will be pink or red now with this counter stain, because remember before at this stage it was clear in color. When we apply our counter stain, it will impart a color of pink or red in those cells. We have to dry these slides now. Either you can leave it upright next to a bunch and let air dry, or we can use bibulous paper, which is blotting paper, and simply place your slide within the booklet, press it gently, not too hard to break the slide, remove it, apply it to a different page, press again. Do this about 3 or 4 times until your slide is completely dried. When your slide is completely dried, hopefully you will see no water droplets or no moisture on the slide. It must be completely dried before you go to examine it under the microscope, because under the microscope, you need to apply a drop of oil and use the oil immersion lens to visualize your cells. All right? And we all know oil and water does not mix. So if you have water here or moisture, it's going to affect the quality of viewing under the microscope. So this is the end of the gram stain procedure. The next stage will be how to examine it under the microscope. This section of the video will deal with antibiotic susceptibility testing, or commonly called AST or sensitivity testing. Now, sensitivity testing is done when, after having done your culture, isolated a pathogen, identified it, and you want to now test that bacteria or pathogen against a spectrum of antibiotics to see which antibiotic you can recommend to the physician in order to treat the patient. So to perform a sensitivity testing, we will need an agar plate called a Mueller-Hinton plate. This Mueller-Hinton plate is a specialized agar used for AST testing or sensitivity testing. We'll need your pathogen, which will be an agar plate. You'll need your antibiotic disc, which we have, a swab for inoculation, and we will now move on to how we actually perform the AST testing. So to do that, you will normally take a flame-cooled loop, which you'll use to pick up your bacteria. Now you'll need around 2 to 5 colonies of bacterial growth to perform your AST testing. So we have our bacteria here. We're going to use a flame-cooled loop, and we're going to pick up colonies. So remember, I explained before what a colony is. A colony is one of these dots, and we're going to pick up just about 2 colonies and inoculate it into some saline. Again, light the flame. Inoculate it. Tap it back. Flame back your loop to make sure you kill the bacteria. And then we have to mix or resuspend this bacteria in the saline to achieve a certain turbidity or standard. Either you can mix it in this way, or you can use a vortex and you'll vortex the tube to get a suspension. Now the strength of the suspension is very important. We have to compare this now to a standard called a McFarland standard, which is a 0.05 strength, or sorry, it's a 0.5 McFarland standard. 0.5 McFarland standard that must be the same density as this. So we'll take our standard tube and compare it to make sure it's the same strength or turbidity. Once it's set, if it's too light, you can go back and add more bacteria. If it's too heavy, you have to decant and add some more saline to the tube to get it to the same density. Once you have adjusted your inoculum with your McFarland standard, you're going to use a milliliter plate again, label it before again with the sample number and the date before inoculated. To inoculate, we'll use a sterile wood applicator or a sterile swab. Open the tube, dip it in, let it wet. While removing, rotate it against the wall of the tube to remove the excess suspension of bacteria. And then we're going to take our agar plate and streak it on. But we're going to use a different streaking pattern here. We're going to put it across one way, turn the plate 45 degrees across one way again, and a third way as if we're cutting a pizza to create slices. After we place that in that fashion, we're going to start from one portion of the plate and streak it by rotating the swab, moving it across the plate without leaving any spaces. Then we're going to turn the plate 80 degrees or so. Do the same thing across the entire plate while rotating the swab. So what we're doing here is basically spreading out the inoculum of bacteria onto the agar plate. And this is done to create a nice constant inoculum so the bacteria is spread out across the plate. This now has to sit and dry for about 5 minutes. Within 5 minutes to 15 minutes, you're going to apply your antibiotic disc, which is here. So to do that, you will need a forceps, which must be flame and cooled. So you flame it. It must be cooled. And you have your choice of antibiotic disc. You will choose your antibiotics. The amount depends on what pathogen you have, what is the expected result in terms of resistance and sensitivity. And that pattern of antibiotic disc will be placed on your plate. Now, there's some commercial systems where you can apply your antibiotic disc in a little dispenser and just press it and stamp it on. It will drop your antibiotics on your plate, or you can do it manually, as in this case. So let's assume 5 minutes would have been passed. You're going to take your cool flame forceps and remove your antibiotic disc one by one and open the plate and apply it onto a spot, carefully without touching the forceps onto the surface of the plate. Just lightly touch it. Flame and cool again. You might want to keep your plate covered during this procedure. Once it's flame and cool again, or use a different forceps that has been flame and cooled, choose a different antibiotic, remove it, and apply it onto your agar plate. Now, once this antibiotic disc has been added to your plate, you cannot move it because the minute it touches on the plate, the antibiotic starts to diffuse out. So if it's dropped in an incorrect place, you leave it there and simply apply another one. After inoculating all your antibiotics, you're going to take your plate now and incubate it, take it to the incubator. Now, notice I added the antibiotic in this direction. When you're incubating your plate, now you have to invert the plate back this way with the agar on top. The reason for that is because overnight during incubation, sometimes moisture will form on the cover And if you invert it the wrong way, moisture or water droplets will fall onto your plate and mess up your reaction. So you want to invert the plate, incubate it with the agar on top. All plates are normally incubated in that fashion with the agar on top. Simply, we will place this plate in our incubator again, overnight, 18 hours growth, 18 hours to 24 hours, and we expect growth the next day. After that day has been passed, we'll remove our plates, and we should get something like this here. This is a small sensitivity plate with Pseudomonas aeruginosa. Pseudomonas aeruginosa has a nice, different species produces nice what they call growth pigments. And some of these pigments, for example, might be pyocyanin, pyruvin, pyroverdin. So you have yellow, green, pyruvin might be red, and so on. So different colors, this bacteria actually shows. So here we have a reacted sensitivity plate with our bacteria, Pseudomonas aeruginosa. And you can see we have two antibiotics this here. On the top one, you can see a clear zone around the disc. Now, what would have happened overnight is that once you place the disc there on the previous day, the drug started to diffuse radially outwards, and the bacteria is trying to grow at the same time on the plate, on the surface of the plate. At some point, you'll see a line or circle or zone created where no bacterial growth occurs. So the drug would have been diffusing outwards, trying to kill the bacteria, and when at some point it will stop killing, you'll have the bacteria growing on the outside. So this is called a zone of inhibition, which we use to measure with a ruler and compare it to a standard chart or reference chart to determine whether it's sensitive or intermediate or resistant. So I'll show you a larger plate now with a similar effect. So here we have another plate. Again, you have several antibiotic discs. Most of them you can see zones of inhibition, all right, clearing around it, whilst this one here, you may see no zone around it. So with this drug, it was ineffective against this pathogen or this bacteria. So you have no you have no zone of inhibition around it. The bacteria grew all the way up to the disc, and there was no killing effect. Whilst the others, you have zones of inhibition or clearing indicating that the drug was effective in killing that bacteria. So here we have another antibiotic sensitivity testing plate where we have different antibiotics. Here we have antibiotic where there's a zone size. Here there's an antibiotic disc without a zone. What would have happened here, because this drug was ineffective, the bacteria grew all the way up to the disc and there's no zone of clearing, whilst in this case here, the bacteria grew up to a certain point and the drug would have killed it again in a circular fashion. So we have zones of inhibition here, no zone here. Again, we compare it to a chart, a reference chart, to see which drugs are sensitive, which are intermediate, and which are resistant. And then the microbiologist will recommend on our report form which drugs that the physician can use for treatment of that pathogen in that patient. And this concludes the sensitivity testing. So the last part we have to talk about for today is the actual reporting from the microbiology lab. So we would have done our initial receipt of the sample, put up our preliminary tests like a gram stain, wet prep, and so on, do our cultures, do our identification, identify the pathogen in question, and maybe perform a sensitivity testing if needed. And now we have to report out those results to the physician on the ward or clinic, etc.